The effect of interval training on the factors influencing maximal oxygen consumption was explored by Michael Rosenblat and colleagues in their systematic review and meta-analysis published in the journal Sports Medicine. Their aim was to review the effect of high-intensity interval training and sprint interval training on the central and peripheral factors that influence VO2 max. This presentation, brought to you by Talking Sports Science, will be a summary of their research. VO2 max is the maximal volume of oxygen consumed, delivered to, and used by the working muscles during maximal effort exercise, and is a key measure of your cardiovascular fitness. Both central and peripheral factors are known to influence VO2 max. Central factors refer mainly to your heart's ability to deliver oxygen to the exercising muscles, which is determined by cardiac output. Cardiac output is a product of stroke volume and heart rate, with stroke volume being the volume of blood pumped out of the heart during each beat. Stroke volume can increase with appropriate training, whereas maximum heart rate is unaffected. While peripheral factors primarily include the ability of your working muscles to uptake oxygen from the capillaries and its subsequent use by the mitochondria. Both capillary density and mitochondrial density can increase with appropriate training. Moving on to the eligibility criteria. Regarding the inclusion criteria, studies were eligible if they included at least 3 interval sessions or were at least 1 week in duration, contained high-intensity interval training or sprint interval training interventions, and involved healthy participants between the ages of 18 and 65, and included at least 1 central or peripheral measure associated with VO2 max. Studies were excluded if the training program was not clearly defined, included high-intensity interval training and sprint interval training in the same program, or included additional exercises that were not quantified, or contained nutritional or recovery interventions, or involved different environmental conditions, or included taking any form of drugs. In terms of the study characteristics, 32 studies involving 320 males and 49 females, consisting of 35 separate interval training groups, met the inclusion criteria and therefore were included in the analysis. High-intensity interval training included 18 groups, while sprint interval training included 17 groups. The high-intensity interval training programs in the studies analysed consisted of work intervals that ranged from 30 seconds to 5 minutes at an intensity between 73 to 100% of peak power output associated with VO2 max, whereas for sprint interval training, work intervals range from 2 to 60 seconds at an intensity ranging from 117% of peak power associated with VO2 max to all-out effort. In terms of the findings, there were no significant differences in the change in VO2 max between high-intensity interval training and sprint interval training. Improvements in the central and peripheral factors influencing VO2 max were dependent on the interval type. Only high-intensity interval training led to a significant improvement in cardiac function. Both high-intensity interval training and sprint interval training increased maximal citrate synthase activity, which is a validated biomarker for mitochondrial density in skeletal muscle, while changes in other peripheral measures, for example capillary density and mitochondrial respiration, only occurred with sprint interval training. Therefore, from a training perspective, performing both types of intervals are recommended. It should be noted that Michael and colleagues do recognise that their conclusions are limited by the lack of trained individuals, small sample sizes, a lack of control groups, and reporting biases among the studies reviewed. And that concludes this presentation. As always, go and check out the full article. The link is in the description. Thanks for listening, folks. See you next time.